Okay, so when we talk of transformation, what actually it means? So transformation is a change from one stage to another stage, or from one form to another form. You might have learned about in entomology about the butterfly, it comes from egg to larva to pupa to adult stage. A transformation from each and every stage to the further stage. A transformation in agriculture in the form of like using the drone technology or the artificial intelligence in agriculture. So in the same way, we use transformation, better to say genetic transformation in plants to change the plant from one form to another form, from one version to another improved version of the plant. So that process is called the genetic transformation. And when we talk about genetic transformation, there are certain methods about that, and we broadly categorize those methods in two parts. The first are direct method, and the second is indirect or biological method. So in the first method, which is the direct method, wherein we directly insert the DNA, or you can say the gene, into the plant without using any intermediary. Okay, like biolistic or particle gun method is there, or liposome method is there. Using the silicon carbide, we insert the gene into the plant or say plant cells. Then microinjection is there, ultrasonication is there, where we use the sound vibrations. Electroporation is there, where we use the electrical pulsage to insert the DNA into the plant cells. Okay, so these are certain direct methods of transformation. So we want to go in detail of each and every method, but obviously we will talk in detail about the biolistic or the particle gun method. On the other side, we have the biological or the indirect method. So here in indirect method, we use certain intermediary, like we use Agrobacterium tumefaciens, which is a bacteria, or Agrobacterium rhizogens, which is again a bacteria. There we insert the gene, and this bacteria will go to the plant or the plant cell and will transfer that particular gene to the plant. So that's why it is called indirect method. Okay. So briefly, we will see both the methods. From direct side, we will see the biologistic method, and from the indirect side, we will see the biological method. Okay. So in this slide, you can see here we have mentioned initially about the vector construct. So vector is a structure of the genetic material which carries the gene of interest or your DNA from outside into the plant. So we insert our gene or DNA into the vector, and then it forms a construct having your gene of interest, and then it goes further into your plant. So we have created a construct here. Okay. After that, we have two options. Either we can go by direct method or indirect method. So if we are going by indirect method, here I have mentioned about Agrobacterium tumefaciens mediated transformation, or you can go by direct method, which I have mentioned here about the gene gun, which I told earlier in the previous slide about the biologistic method of transformation. Okay. So in the both the method, what happens? We take certain explant. Explant is a certain tissue of the plant which is used for transformation. You can take the leaf, you can take the seed, you can take the embryonic axis, you can take the cotyledonary node, apical meristem, or even if we take the half seed also. So what happens? Like we transform these explants, okay, using any of these methods, and after transformation, we give the selection pressure. Now, the selection pressure depends on that while inserting the gene into the vector construct, okay, what we did, uh, either we have inserted some gene which is providing the resistance to some antibiotics, or we have given a gene which will give some colorimetric or color effect if it has been transformed. Okay, so in general, the selection pressure we give is of the antibiotics. Okay, so if that gene has been transformed against the which works against what you call for the antibiotic selection, then your explant will survive. If those genes have not been transferred, so if you expose it to the antibiotics, obviously that won't survive. So a selection pressure has been given here. After we give the selection pressure, then we go to the next stage, that is regeneration. From those explants which have been transformed by our gene of interest, what we do now, we will regenerate the shoot. Okay, the first shoot is generated, then root is generated, then it is acclimatized. Okay, after that, what we do the molecular screening of the transformed plants. So molecular scanning in general, what we do the PCR, polymerase chain reaction, amplification of the gene of interest. So we take the primers for that gene, and we see that either those genes have been inserted into the plant or not. Okay, so that is the molecular screening. Once that has been done, so those plants which are positive for that molecular screening, that is further moved for development, and then field trials will take place, and other procedures will be followed for its release and all those things. So this is just a gist which shows that whichever method you follow, this is the mode by which you have to develop a plant if you are going for the genetic transformation. So here first let's talk about the agrobacterium based transformation. So agrobacterium is a bacteria. You might have seen, or many of you might be knowing about the crown gall disease in the plants. Okay, most of the dicot plants are having the crown gall disease, wherein the agrobacterium infects the plant, and a gall, means a tumor type structure, will be formed into the plants. In many of the plants, like in mango, you might have seen a swollen portion somewhere here and there. So that is just a crown gall that occurs due to the infection from the agrobacterium tumefaciens. So since this bacteria has the natural ability to infect the plants, so this particular bacteria has been exploited by the molecular biologist to transfer the gene of interest into the plant using its own machinery, through which they transfer their own gene into the plant in a natural way. So, agrobacterium is having a TI plasmid, okay, that is tumor-inducing plasmid. Because crown gall disease, also known as, it is just like a tumor. So that's why it is known as tumor-inducing. So in that plasmid, you have the Vir region. Vir region is actually a virulence region, which helps in infection of any of the plant which they infect. So this region has in total 8 operons, and generally 8 genes express themselves, which further helps in proper infection of the plant. So if you see here, so this process takes place in broadly in 6 major steps. First stage, signal recognition by agrobacterium. So what happens? Any plant which is wounded, okay, in nature, or whenever you do the genetic transformation, so if the plant is wounded, it will release some chemicals, sugars, and specifically, if I would like to mention, a chemical named acetylsalicylate is there, the phenolic compound. So that compound is sensed by the agrobacterium. Okay. So here the Vir-A gene, you might be saying in this picture, the Vir-A will sense the acetylsalicylate or the phenolic compounds, and Vir-A will activate the Vir-G by phosphorylation. It will consume the ATP by phosphorylation. It will activate the Vir-G. Now Vir-G, what it will do? It will go and it will activate the entire Vir regulon. All the Vir genes will be expressed further, including the Vir-A. So now what happens? Virg activated Virg activated all the Vir. Now, here, what happens? You can see the left border and the right border. Okay. So, between these two LB and RB, your gene of interest or the transfer DNA has been inserted. Okay. So, the Vir D1 and D2, this complex will cut your transfer DNA or your gene of interest, whatever you wish to transfer, which has been loaded between these border points. So this will be clipped from these 2 points by Vir D1 D2 complex, and the single stranded release of tDNA, a single stranded form will only be transferred to the plant cell. Okay. So at the 5' end of the single stranded tDNA, the VD2 will do the capping to protect it from further cleavage. Okay, so VD2 has what it has done, it has cut also, it has coated itself on the 5' end also, and now this VD2 is having the nuclear localization signal also, means it will lead this single stranded tDNA transfer from agrobacterium to the plant cell nucleus also. Now what happens here? Another compound is there, that is VE. VE, what it will do? It will form a coat on the single stranded tDNA to protect it from what you call cleavage or degradation. So VIR-D2 and VIR-E2 will form the coat, and a single stranded tDNA will move from the agrobacterium cell via the channel made by VIR-B. You can see here a channel has been made by VIR-B. So through that channel, it will move to the plant. So now in plant, your single stranded tDNA has moved along with the VIR-D2, VIR-E2, and another factor is there, that is VIR-F complex. So VIR-F will also move along with this complex, and now it will go into the plant cell, and there through the nuclear pore complex it will enter the nucleus, and there it will do the illegitimate recombination. Means the recombination will not be guided one; it will randomly go and insert into the plant DNA. So this was about the agrobacterium based transformation. So in this slide again, I have shown the same procedure in a broad way. Like you took the explant, you did the pre-culture of the explant, then agrobacterium infection was given, and then it was co-cultured with the agrobacterium. The explant was co-cultured with the agrobacterium. Then agrobacterium inhibition has been done. Like after the co-culture has been done for 2 to 3 days, a chemical named generally what we use is cefotaxime. So that cefotaxime chemical, it will kill all the agrobacterium, all the live agrobacterium, because already the gene transferred has taken place. So now the live agrobacterium is no more required. So the agrobacterium is killed by those chemicals. And after that, it will go for selection and regeneration. And again, it will form the entire plant. On the other side, what you can see here, in the tDNA structure, we have given some Gus gene also. Okay, so through the colorimetric test, you can check either your gene has been transferred here or not. Here you can see using the uh, primers for your gene of interest, we did the PCR, and we made it sure that your gene has been inserted into the plant or not. After these molecular detection only, the plant is further propelled, processed further for the development and further checking and further release and all those things. If anything is there that means I'm going fast, or if something has to be made it more clear, please let me know. So
Okay,
let
sir.
us talk about the another mode of transformation. What we talked earlier, direct and indirect. So agrobacterium was the indirect mode of transformation, and now we'll be talking one of the method of transformation, that is direct method of transformation. And there we have taken the biologistic method. We are taking agrobacterium and biologistic in detail because these are the two major transformation methods which are generally used in most of the laboratories, and that's why I'm talking about that one only. So ballistic method is also known as gene gun or particle bombardment method of transformation, or it is known as the ballistics because just like the ballistic missiles, what we use in the same mode it has been created, and therefore it is known as the biological ballistics. And ballistics is the short form of the biological ballistics. Okay, so it is also known as the particle mediated gene transfer. So this method has been generally used almost since like 1986 or 1987, and even now in most of the laboratories, this has been used for genetic transformation of the plants. Okay, so as I mentioned, in 1987 it was introduced, this method, and in 1987 it was used on onion tissue, and this method actually came after what you call the first transgenic tobacco plant, which was created in 1983. So after that, this method came. So 1983 transgenic tobacco that was created using the agrobacterium mediated transformation only. And in 1993, the first transgenic sorghum plants were also successfully regenerated using this method. So, what is the principle of biologistic method of genetic transformation? So, here basically what we do, uh, DNA coated microprojectiles, okay, are accelerated and then it is targeted on the target cells. Okay, we will see in detail in subsequent slides. So, a few major components of this ballistic method is helium gas, then particle carriers, which are the microprojectiles. Microprojectiles are generally made of gold or tungsten because these are the inert materials. Gold and tungsten both are inert, and therefore uh, these particles are being used where on which your DNA is coated and then bombarded on the plant cell. Okay, and that particle goes inside the plant cell and will transfer your gene of interest or the DNA into the plant. Okay, and therefore, what you call since gold and tungsten both are inert, and therefore it doesn't cause any harm to the plants. So let's have a overall view of the particle bombardment. Here, DNA coating is when done. Okay, so DNA is precipitated using calcium chloride, and spermidine is used, which stabilizes your DNA. Okay, and it is coated onto the small metal particles, which I have already mentioned, that is gold or tungsten. Okay, and a pressure helium gas chamber is there. Okay, a pressure is created from the helium gas chamber, which propels your what you call DNA coated particles. Okay, it propels with a high speed, and then it will go and bombard itself on the plant cells. Okay, and thus the genetic transformation occurs. So here in the image also you can see like a pressurized gas, the gas with a high pressure has been created. Okay, a bullet has been shown here. The bullet is actually a macro carrier, which in this here they have mentioned it as a bullet, but it is a macro carrier wherein the DNA coated metal particles has been attached there. Okay, and here is the rupture disk. So whenever the helium gas pressure will go beyond a certain limit, so that limit actually is defined by the rupture disk, that how much pressure it can handle. So once the pressure goes beyond that limit, that disk will rupture, and the high pressure helium gas will propel the bullet or the macro carrier, and it will go further. And here you can see the filter screen. Okay, so here it will stop and it will discard all the micro particles further, which will go and insert into the plant cell. Okay, so I think this is clear. So in this slide, we can see in a better way. Okay. So here you can see a gas acceleration tube is there. Okay. Here you can see the rupture disk. So whenever the pressure will go beyond the capability of this rupture disk, okay, this will burst. Okay. And once it will burst, this macro carrier with the micro projectiles, okay, will go further and it will be stopped by the stopping screen. Okay, so this macro carrier will not go up to the plant cells. This macro carrier will be stopped by stopping screen, and once it is stopped, then this micro projectiles get separated from this macro carrier, and it will go and hit all these target plant tissues. So here you can see the gas has ruptured the rupture disk. Macro carrier was stopped by the stopping screen, and these particles it was propelled further up to the target cells. Okay, so this is an overall procedure for doing the ballistic mediated method of transformation. I think much of the facts we have already talked here, so I think no need to repeat the same thing. So here I mentioned about the helium pressure, that is 1200 pound psi, pound per square inch. So that I was mentioning, like this pressure depends on the use and the capability of the rupture disk and how much pressure you want, which type of cells you are using. Okay, so upon that it can vary. Okay, so rest of the things have been already covered. Okay, so talking about the applications of ballistics technology, so all the applications are same as it is for the genetically modified organism or for genome editing. Okay, like we can do plant genetic engineering, we can do animal cell transfection, vaccine development, you can do go for gene therapy, you can deliver the genome editing reagents into the plants. Okay, now, one point I mentioned here that if you compare with the agrobacterium, so gene gun is especially useful when agrobacterium mediated transformation is not effective. Like in originally it was being used for dicot plants and it was not so working well for the monocots, but nowadays it is even working for monocots, but still it is not that effective. And therefore, using the ballistic method or gene gun, it eliminates that limiting point of the agrobacterium. Okay, so irrespective of whatever the plant or the cell you are taking, gene gun is totally effective compared to agrobacterium. So host limitation is not there. So advantage, talking about the advantage of ballistic method. So what I mentioned earlier, it circumvents the host range limitation, okay, which agrobacterium has. Then many different tissues and cell types can be transformed. It's a direct DNA delivery. Multiple plasmids can be integrated together for the core transformation. It is simple, and even large DNA fragments can also be delivered into the plant. So it can be used even for RNA or protein also, less labor intensive. Okay, and again in many of the cases you can reduce the amount of the plant tissue culture involved in transformation compared to the agrobacterium. Again, every technology has its own limitations, and same stands for the gene gun method also. Like in a few of the cases, efficiency is low because your cell how much permeable it is to the ballistic method. So again, it varies from cell to cell or tissue to tissue. So that efficiency matters a lot. Then again, since we are bombarding the microparticles with a high speed, so again, it creates damage to the cells. So once a damage has taken place to a significant extent, regeneration will not be possible from those cells. So the equipment is very expensive. So the cellular target cannot be controlled. It can go to the cytoplasm, nucleus, mitochondria, plastid also. Because in agrobacterium, what we saw in agrobacterium, nuclear localization signal NLS was there, which was provided by VD2. But here that is not there. So along with the nucleus, it can go to cytoplasm also, or it can go to mitochondria or the plastid also. So this in summary about the plant transformation method. So I just few slides about genome editing because I thought if someone has not talked about this genome editing, I'll just have a few slides on that one. So I won't take much time, hardly 5 minutes more. So while talking about genome editing in plants, we all might have uh, heard about the CRISPR Cas system because genome editing has become a buzzword nowadays. So in genome editing, there are various methods through which you can proceed. And to mention few like the zinc finger nuclease, talents, and then CRISPR. And CRISPR is been most extensively used these days uh, in most of the laboratories. So what actually happens? See, DNA is there in each and every organism, in each and every cell. So breaking of DNA and further repair, it happens in nature. Okay. So the break and further repair. So a gap between the breakage and repair. So through genome editing, we have been interfering at that point and we have been exploiting that machinery as per our need in the plant. That breakage, we do the breakage and then further we direct the repairing of that particular part of DNA to achieve the DNA what we desire. Okay, to create the specific outcomes. So what actually happens? Like if you talk of CRISPR-Cas, so it may either delete something, it can edit also, it can replace also. Okay, and for n number of traits you can use it, like for better nutrition, longer shelf life, abiotic and biotic resistance, and higher yield, and moreover the time required for developing a particular what you call plant or the plant type it reduces in compared to the conventional plant breeding. So I was mentioning about the break and repair. Okay, so what happens? Whenever there is a cut into the genome segment, so cut is being created by the nucleases. We already know. Okay, so after the cut, the repair will take place either by homologous recombination or it will take place by non-homologous end joining, and it happens in nature too. So what we do whenever there is a non-homologous end joining, okay, so there will be loss of few base pairs or there may be addition of few base pairs too. Okay, so in general what happens since it is not directed one, okay, so these are mostly error prone. Okay, so in general what happens, these small insertion deletions, it in general inactivates the gene. Okay, now on the other side, so through this also we are exploiting the CRISPR-Cas9 technique for our need. On the other side, where homologous recombination takes place, there we give our DNA template, whatever the insertion and deletions we need it. Okay, so that is directed, and thus insertion or repair takes place, and we get the desired result. So CRISPR system actually it has been taken from the bacterial system, okay, where the bacteria or the prokaryotic system use it as a defense mechanism, okay. There they take the parts of the virus attacking those bacteriophage or the virus attacking those bacteria, and they will integrate it into their genome, and they will form the tracer RNA. And this series of the parts of the genome of the virus into the bacterial genome will further form the tracer RNA on expression. And whenever the further what you call further infection by the bacteriophage will take place, this tracer RNA it will go and it will destroy that particular virus. Okay, and this mechanism was actually used, and our genome editing tool has been accordingly designed, exploiting this system. Okay, so these are the two scientists, Emmanuel Carpentier and Jennifer Doudna. They got the Nobel Prize for developing the CRISPR-Cas9 system, Nobel Prize in Chemistry 2020. So here are the few what you call applications of plants. I already mentioned that different biotic stress, abiotic stress, yield improvement. Even it can be used for anti-nutritional factors, biofortification. Then understanding the genome. Understand the genome means you can by knocking out or by creating some gap into any gene, you can know the functions. Okay, so understand the genome. And the most important as there was a lot of hue and cry regarding the GMOs and all those things. So this is non-GMO crops basically, because there are three modes of creating the genome edited crops in general. That is known as SDN 123. SDN is site-directed nuclease. So what happens like in SDN 1 and 2, few base pair changes takes place. Okay, in SDN 1 template is not required, in SDN 2 some small DNA template is been given for some specific changes to take place. But in SDN 3 what happens, a large DNA elements or full length genes of foreign origin also been integrated into the genome, unlike SDN 1 and 2. And therefore the government of India has given permission for SDN 1 and 2 only in 2022, and not for SDN3 to be released into the environment or to be released for the farmers. So in SDN 1 and 2, we are not bringing any foreign DNA or foreign gene from any other organism. We are doing certain changes into certain base pair changes into the DNA or the genome of the plant. Okay, that can be random. If you want to knock out some gene, that can be random. Or if you want to correct certain gene, like one base pair correction can improve a certain trait of the plant, so that can be done through the SDN2, that is using a small DNA template which will direct the precise correction into that particular location of the DNA. Okay, so this SDN1 and 2 has been approved, but SDN3 has not yet been approved. So, taking few examples, that is one is GABA tomato. There's gamma butyric amino acid. Okay, so that are the first gene edited product through CRISPR-Cas9. So the high GABA actually, while when the tomato goes for ripening, that GABA content is reduced in the ripening process. So through this mechanism, high GABA content was maintained in the plant, and this is helpful for reducing the blood pressure and promote relaxation. Then biofortified tomatoes, wherein vitamin D was enhanced. Then mutations in the promoter of sweet 13 genes in rice created the bacterial blight resistance through genome editing. And powdery mildew resistance in wheat has been created by knocking down the susceptibility locus MLO, that is mildew locus. That is a susceptibility gene which, when uh, activated, it will make it susceptible to the powdery mildew. And therefore, knocking out this particular gene, what it will do, it will make it resistance. So these were the few examples. And since I was about to talk on transformation only, so these slides just I kept for a brief, just superficial introduction to genome editing. So that's why I just kept 4 or 5 slides. And I think that was all about from my side. Thank you.